Okay, so let's get started. Hello, everyone. Welcome to our today's museum seminar. Um, it's Thanks everyone to join us today. It's absolutely my pleasure to welcome our today's speaker, Daniel Pinschewer from Switzerland. So I actually read some of Daniel's work because I'm working on mucosal immunology, and some of my work is related to the biology. And then some of his work has really blown my mind away. So I think it's really great for us to learn his science today. So Daniel actually completed his MD training in Switzerland in University of Zurich, and then afterwards he briefly did his postdoc in Scripps Research Institute La Jolla in US, and then shortly followed with another postdoc in University of Zurich. So from 2004, Daniel started his independent research program as a group leader in University Hospital of Zurich. And then afterwards, he was promoted as associate professor to, and then relocated in University of Geneva. From 2013, Daniel actually moved to University of Basel as a full professor in Department of Biology. So Daniel's research is mainly interested in the interplay between the persistence in microbials, both virus and bacterial, and their mammalian hosts. His research emphasis is mainly on the host immune defense with special focus on the B cells and antibody responses, vaccines, and then alarmins. In long term, his lab investigated the different aspects, which including B cell defense in persistent viral infection, B cell responses in antimicrobacterial immunity, and then B cell responses in persistent microbial infection, role of alarmins in T cell immunity, and virally vectors vaccine development. So therefore, his research actually covered both adaptive and innate immune defense with persistent microbial infection as a common theme. His lab actually combined cutting-edge mouse infection models, high-end cellular immunological techniques, genomics, and then aboard a range of molecular analytics with molecular biological techniques for the engineering of infection virus. And then his research holds a promise for clinical translation in the context of the vaccination and treatment of persistent microbial disease such as chronic viral hepatitis and then tuberculosis. So I think it's really exciting to have Daniel on our platform today, and I think he's going to talk about immunity and tolerance in the viral infection to us. So thank you so much, Daniel. We are very excited to listen to your seminar.
Thank you very much, John, for the generous introduction and specifically, for the platform, for inviting me. It's a great pleasure and honor to speak today. And I hope that, some of what I'm going to present is going to be interesting and helpful to this audience. You've said that. Don't need to repeat this. Just a general overview of my lab does. And just to admit that we're mostly working on mice. So we're really trying to understand how mammalian systems, mammalian multicellular organisms, try to fight off, deal with, or get sick from viruses and persistent bacteria. So for today, I chose two chapters, separate chapters that really don't have very much to do with each other except for some model-related elements. One is recently published, one is unpublished. And the first one is about the durable depletion of lymphocyte subsets based on AAV-based technology, which helps us to dissect immune control of chronic viral infection. And the second part is going to be about tolerance or the limitations of tolerance, the limits in so-called viral carriers, meaning lifelong persistently infected animals and supposedly humans. And that relates more to human hepatitis B, and such diseases. Good. I'll go right into matters and introduce you to my favorite long-term used model, which is lymphocytic choriomeningitis virus, or abbreviated LCMV, that probably many of you have used or heard of. It's widely used in immunology and has been used for decades, almost a century in immunology. And there are different combinations of mouse strains and virus strains and dosage and route of infection you can use. And oftentimes we like to differentiate an acute infection model from a chronic infection model. And in acute infection, we're speaking of a relatively short-term viral load curve, which is eliminated below detection limits within about a week. And this is achieved by a very vigorous CD8 T cell response, and hence also the wide use of LCMV as a model to trigger and study CD8 T cells. There is also a chronic model where you inoculate the animals and then virus goes up, but it for a long time stays up. And it can easily take 2 months or a bit longer until virus comes back down below detection limits. We like to call this chronic. It is not persistent, if you wish, but it mimics a little what we see in the successful clearers of hepatitis C, for example. So those people have viremia for 2-3 months, but then get the upper hand over the virus. So I think it's an interesting model because we like to understand how this is functioning, and possibly how we can tweak it so that those that turn chronic with such infections would also go the self-limiting protracted viremic process, such a curve of declining viremia and eventual elimination. So what is the mechanism of that late control? What's the even correlate that allows the mouse, in that case, to eliminate the virus? We know that B cells are important in some way. CD8 T cells, as I said, are sort of historically known as important players in LCMV infection. And we wanted how to best study their individual contribution to that process. How to eliminate B cells, CD8 T cells from that game. And the protagonist of that study is Ina Kastner, who completed a PhD with me a good year ago and now moved on to industry. She's done a terrific job, and I'll share her slides and work with you in the first part of the seminar today. So as you might say, well, it's easy. We use knockout mice, so genetically engineered mouse models, and that works. So if you take a B cell knockout, B cell counts will be strictly 0 over the entire course of the lifetime of the mouse and of your experiment. However, generating a new model or so can take a lot of time and can be costly. You can also use depletion antibodies, and I'm sure most of you do in your experimental work. So you administer recombinant antibody or haploidoma-produced antibody to a mouse, and then depletion antibody eliminates your cells, but it will wash out. So you see this would be antibody concentration as a function of time. It washes out, and as it washes out, its effect goes away. So your, let's say, T lymphocyte count, depleted lymphocytes, they go down, but as the antibody washes out, your cells come back. So it's a transient effect. You might say, well, it's good enough for my experiments. Perhaps if you do week-long experiments and use on T cells, that's perfectly fine. If you use neutrophils and you want to work on them for 2 months, it's going to be extremely hard. You will have to re-administer, the multiple syringe, your antibody very regularly. It will take you as an experimentalist a lot of time, and probably the mice don't appreciate very much being injected like 2 or 5 times a week. So the question that Lena asked is there a way to permanently deplete without genetic modification of the animal, germline, and without repeat administration for the entire course of our infection model, which is a good 2 to 3 months? Now, you might be familiar with AAV technology, or adeno-associated viral vectors, which is widely used in gene therapy these days. So illustrated down here, we've got products on the market, but these delivery vectors, so viral vectors, have also been shown to express, for example, here in mice, very durably antibodies that are encoded in the vector genome. So that's a study from 2011, and we've done that with antiviral antibodies. As these people, this was an anti-HIV antibody administration study. So we wondered, could we, instead of putting an antibacterial or antiviral antibody, could we put a depletion antibody into the AAV? So this is the idea, right? You have got this AAV genome, and you can encode heavy end light chain of your antibody of interest in that AAV genome. You would then administer the AAV to the mouse. And what happens is that the AAV is given intramuscularly. It will go episomal in the muscle cells that it infects. And it will stay in these muscle cells with a half-life that is very, very long, and just keep producing antibodies from the muscle. So wrong tissue, I get it. Completely wrong tissue, but in the end, it's an antibody. And the expectation is that the antibody titers would rise quickly and then just stay stable because it's continued supply from the muscle. And if this is a depletion antibody, would hope that these antibodies bind to your lymphocyte subset of interest, and then the count drops but stays low. So it would not rebound when the antibody is washed out, because it would never wash out. There would be continuous production. So Lena did some proof of concept experiments. I'm showing you only one example here, anti-CD4. Anti-CD4 antibody, very widely used. You can give a single shot of the antibody, it gives you a certain serum titer of the antibody, then it washes out over a good month. She gave an AAV encoding the same antibody, titers even higher, but they stay high for over 100 days. This is what the CD4 T lymphocyte population does in blood. This is the reference animal without any depletion, stable population of CD4 cells in blood. Then you've got the animal with a transient passive antibody administration, also depleted very nicely, but then after a month, when the antibody washes out, CD4s come back. Those given the AAV vector expressing the antibody are undetectable. I'm showing this here. So after 100 days, the transient depletion animal has a very nice population, pretty comparable to the control treated. The AAV anti-CD4 treated has no CD4 cells left, absolutely none. So, several orders of magnitude below our detection limits. Okay, so proof of principle, it works. We can use AAVs for long-term depletion after single administration. Just to emphasize that again, we give that AAV once, and it sits in the muscle and makes the antibody for the rest of the time. We don't have to inject the mice a second time. We developed a whole toolbox. I'm just showing some of the constructs. We've got AAVs to deplete CD4, CD8s, B cells, for NK cells, which are hard to deplete long-term, or neutrophil, the worst. We've got other constructs. We also, and I'm not showing this today, for example, have antibodies or decoy receptors for cytokines. So you can do a cytokine knockout, but starting in adult life of the animal. It's pretty versatile and interesting. And if you're interested in using some of these tools, you can order these vector genomes or also the infectious ready-to-use vectors through the European Virus Archive online. So are the depletion AAV advantages? Why, besides the durability, why should we or would we be interested in using these tools? Well, first point is that you can long-term deplete any mouse. So if, for example, your favorite animal model is an FVB or is a non-black 6 strain, make it New Zealand white or New Zealand black or whatever, it works. I'm not showing these data. They're published, so you can look them up. You don't have to back-cross your genetic modification to your genetic background of interest, which is very tedious. But you can do your sort of somatic knockout in adult life on the day you choose your model. Then there are also deficiencies of genetically modified mice that we don't like. So a famous example is the B-cell deficient mouse, which is widely used, but it not only lacks B cells, but it also has secondary defects. For example, the marginal zone is, and I'll show this in a moment, is severely hampered in these mice. So when your B-cell deficient mouse has a biological phenotype, you cannot be totally sure it's because of the B cells. It might be because of the accessory deficiencies. And we wondered whether B cell depletion by the AAV, which is after the ontogeny of the animal, would prevent these accessory deficiencies from taking place. So, for example, would that preserve the marginal zone? I'll show you what I mean first. So this is a regular wild-type black 6 mouse. It has in histology these nice B cell compartments, B zone. It has CD169 positive macrophage around the white pulp follicle that you see here. It has also some SANA1 expressing macrophages in that area, and it has the red pulp macrophages. Now, if you go to a JH knockout or a MUMT, would be the same. The widely used B cell deficient mice, yeah, they don't have the B cell compartment. But if you look out for the marginal zone, I mean, it's not much to find. This is virtually absent. So you lack the marginal zone mantle around the white pulp follicle. It's not there. But if we deplete our mice with the AAV anti-CD20 so that we also get rid of all the B cells, you've got a beautiful preservation of the marginal zone macrophage compartment, so by both markers, which means that it was sufficient apparently to have the B cells there doing the mouse ontogeny, and then the marginal zone will, at least for the 45 days of study here, will be preserved, which allows you to more specifically ask, what is that B cell really doing, as opposed to all the other defects that you encountered in the knockouts. So yes, unperturbed ontogeny of lymphatic structures is an advantage. And last but not least, I mean, who has not intercrossed knockout mice or transgenic with knockouts or so to combine defects? I mean, we have oftentimes. So we asked, can we co-administer 2 AAVs and deplete 2 subsets at once? And yes, that works beautifully. I'm not showing the numbers, but just the histopics, I think it says it all. You've got control mice that have B cells, CD19 positive, and they have CD8 cells. Then you have animals that were given AAV anti-CD20 to deplete the B cell compartment, it's gone, or those that got the anti-CD8 AAV, they have no more CD8 T cells, or those that got both AAVs and then both compartments are gone. So it's very versatile. You can combine them and avoid the month and costs of crosses. Okay, so that works too. Now, besides the toolbox, this was not just a tool development effort. It was really meant to be used for us for investigation of the immune control in chronic infection. That's what I started out with. So what's the control mechanism? And we started from the CD8 and B cell side and said we know from all published literature, including ours, that they play some role in one or another way, but how exactly they synergize and interdepend and so on is unknown. And a first experiment was really trivial. You have viremia of a control animal. So you see it starts very high, but then over the course of about 80 days, it goes below detection limits. So is that clearance, so this protracted long-term chronic clearance, is that preserved if you remove, for example, CD8s? So animals were given, prior to being infected, the AAV anti-CD8 construct. And what happened? Well, the initial viral load drop was maintained. So the antibodies apparently did their job and suppressed viral loads. But then virus rebounded. So apparently CD8s are needed here from about, 60 to 80 days onwards to clean out what is left. So what happens if you remove the B cells from the start, but preserve marginal zone, which was an interesting experiment to do for us? Well, here there's not much of viral load control visible. And just to show you the B cell knockout animal, we included that in the experiment too, is pretty much identical to the CD20 AAV depleted animal. So these two curves, they are overlapping. We can't discriminate them. So it's a good reflection of what the B cell knockout mouse does. And there's apparently loss of viral load control, complete loss of viral load control when there's no B cell around. Last but not least, we combined now the CD8 and CD20 AAVs. And my expectation, I told Lena ahead of the experiment, was that there would be even higher viral load. And I was very disappointed. There was no difference. So you see, this group here, the double depleted, is as good or as bad as the CD20 depleted, meaning that the combination depletion doesn't make it worse. Once there are no B cells around, CD8s seem to be useless in that setting. So in summary, B cells are required for viral clearance. There's transient viral load control without CD8s, but when B cells are absent, CD8s seem to completely fail to suppress viral loads. Now you could say, well, fair enough, but maybe the B cells do something else than just directly suppress virus. They might also play a role in the T cell responses, and the T cells might do the job. So we tested that, and we looked in the earlier phase of chronic infection, when viral loads in these various groups are still comparable, for potential effects of depletion on T cell compartments. So the same groups as the previous slide, and without going all over these plots, this is specific CD4 cells to LCMV. We found that depleting B cells, so here are the numbers, or here, profoundly reduces CD4 T cell responses. So the animals were not just devoid of B cells and antibodies, but they also had difficulties mounting a good CD4 response. And here the T follicular helper response is virtually absent in these animals. The same for depletion as for knockout. With CD8 depletion, that doesn't happen. So CD8 depleted mice make beautiful CD4 responses, but the B cell depletion profoundly influences and reduces CD4s. We did look at CD8s as well and had an analogous pattern. So B cell depletion or knockout reduces CD8 responses. And that brings us in a somewhat difficult situation. So the impaired CD4 and CD8 T cell responses when we remove the B cells. So is it now chicken or egg, right? That prevents viral load control. You could say there's no viral load control when there are no B cells, but there are also impaired antiviral CD4 and CD8 T cell responses when there are no B cells. And both or either one of them might directly or indirectly affect viral load control. So what is what? A direct effect would mean that B cells make antibody and that antibody acts on the virus. The indirect effect would be that the B cells in some way or another influence the CD4 and CD8 response, and we showed that is the case. And it's actually the T cells which affect viral clearance or mediate viral clearance. So trying dissecting this, we turned to some sophisticated knockout models again. Here the AAV technology comes to its limits, and we used a mouse that has a diverse B cell repertoire but cannot make secreted antibodies. So this is double knockout for soluble IgM and AID, meaning these B cells have a membranous IgM receptor and IgD, but they cannot class switch and they cannot secrete the IgM they have on the surface. So they make a reasonably normal B cell response in the absence of antibody. And that allows us to see to which extent the antibodies influence viral load control. Secondly, we used a monoclonal B cell receptor mouse, which has a B cell receptor uniform in all B cells that is irrelevant for the virus we're using. So it's specific for a completely different virus. Therefore, we have a monoclonal irrelevant repertoire of B cells. They can make antibodies, unlike the ones on the left, but the antibodies are useless in the present context. So we used these mice, and for further control, we used B cell depleted wild-type mice and checked for CD4 responses. And that again, was a big surprise. So my prediction were completely off. What we found was that the presence of B cells, irrespective of whether they can make antibodies or whether they have a useful receptor, restores CD4 responses to completely normal levels. So this is the control. This is the B cell depleted, 1 log difference. But when you have B cells around, no matter what they can secrete, no matter what specificity they have, they're at least equivalent to the control. And the same for T follicular helper cells. CD8s, same picture. So having B cells around, no matter what they can do or what specificity they have, your CD8 response is normal. So the physical presence of B cells is required for unimpaired antiviral CD4 and CD8 responses. But that role of the B cell is independent of the cell's specificity or ability to secrete antibodies. Okay, so that gives us a tool to finally ask the question, how do B cells contribute to virus control? So we used these mice now and checked virus load over time. And you see, again, the wild type animals, they clear out the infection over the 80-day period, but all our depleted and modified mice are persistently viremic. So if they have specific B cells, but cannot secrete antibody here, virus load is not controlled. If they have antibodies, but wrong specificity, here the triangles, viral load is not controlled. So specific antibody responses are indispensable for resolution of chronic viral infection. If I summarize this, what we learned from depletion antibodies delivered by AAV, which I think in the present context of an 80 to 100-day experiment was the only way we could do this, is that in a wild-type animal, you see this protracted clearance. In an animal that has been rid of its B cells, virus persists. If CD8s have been removed, virus is transiently suppressed and rebounds. And if you remove B and T cells altogether, you can't make things worse. And in terms of the actions over time, we think that the first actor is the B cell making antibody, and the antibody is really the antibody, not the B cell. They suppress viral loads to a certain point, maybe around here in my curve, where the rebound would happen if there were no CD8s, and then CD8s come in and sort of clean out the rest, and there, you end with a resolved infection. The B cells are required, just physical presence required for normal CD4 and CD8s. And of course, CD4s are required for a good B cell response, and perhaps CD8s, but we haven't seen this in our work. So it's really a good team effort that clears out infection, but we really see B cells as the first actor here and the most important actor, although, we and many other labs have extensively studied CD8s in the context of chronic L-simple infection. Would somebody like to ask a question to this part of my talk before I move on to my second one?
Well, I think you can move on, Daniel.
Good. All right. So second one is about tolerance. And I'll give you some background and then some history of the question first. So, I've been talking to you about this model where you give LCMV to an adult mouse, and then you follow it and see how it deals with a virus. And you can find that virus is normally controlled, and there's a very vigorous immune response. You saw massive antibody, CD8, CD4 response, and so forth. But this infection model is not what the virus actually has evolved to do in its evolution. It's naturally transmitted from mother to offspring, and thereby is handed over to an animal which we have learned is tolerant to the virus. So we can mimic this in the lab by giving the virus to the freshly born. So within the first 24 hours, we regularly control the animals. Once the dam has given birth, we inject animals right away. And thereby we establish a carrier status. So these animals have virus from head to toe. They are perfectly healthy. And they have specific tolerance to the antigens of the virus. And that is analogous to hepatitis B carriage in humans, or thought to be analogous, which is a globally very important medical problem with about 300 million carriers. So again, supposedly tolerant individuals, immunotolerant individuals. Again, I said these animals are full of virus from head to toe, and they're not mounting a classical immune response as you would see it in the adult infection. And this observation, that there is virtually no immune response, this dates back to the 1930s and has very much stimulated the concept of tolerance over the decades to come. So this monograph that I very much recommend everybody to read by Burnett and Fenner from 1949 cites, this is sort of the beginning of the clonal selection theory, the idea that there might be specific cells that multiply and thereby can carry, ascertain the diversity of the immune response. This monograph suggests that by consequence, the individual would learn in its ontogeny what is self, and then eliminate the clones that react to self, so that it would be self-tolerant. So that was proposed in 1949 by Burnett and Fenner, and we learned by experimental work carried out in the late 1980s with transgenic CD8 T cells that this can happen. So when you have LCME-specific CD8 T cells in such a newborn animal, they will get deleted if virus is administered in the neonatal period. And later on, the same has been shown for CD4 T cells. So the concept seems to hold. There was, however, the long-standing observation which somehow didn't fit. And that's a 1967 paper by Mike Alston and Frank Dixon from Scripps, where they reported that these tolerant virus carriers produced antibodies specific for the virus. As you may know, Burnett has been awarded the 1960 Nobel Prize, not alone, but together with Peter Medawar from University College London. And Medawar's lab did the experimental work to prove that neonatal exposure, they used allogeneic cells, rendered animals tolerant. So they also experimentally proved that seeing the antigen in neonatal time, or even intra-utero, would render the T cell system tolerant. Importantly, however, they always looked at CD8s because they looked at lytic units, so killing assays back in the days. Work conducted in the same tolerance model in the 80s showed that there was some supposedly helper function. So there was a B cell response to the foreign cells in these supposedly tolerant animals. So this is, a wrap-up of the experimental work, but I'll show you why I'm interested in this. I'm very much interested in this because of Hep B. So hepatitis B carriers, they have this phase of tolerance where there's no liver inflammation, and it's called immunotolerant phase. There's no liver inflammation, sky-high viral loads. I mean, it's really a walking incubator of hepatitis B. These patients, they're completely healthy. But then most of them, after 20, 30, 50 years, progress to a so-called immunoactive phase. And the immunoactive phase is characterized by fluctuating viral loads, which typically are at least partially suppressed, and they can eventually progress to a low replicative phase, in which the patient is through the worst of the disease. Viral loads are very low. They're not eliminated, but very low, so partially under control. And we do know by simple correlation that the immunoactive phase is characterized by a rise of antibody specificities to several determinants of the virus, but the causal link, if any, is unknown. And it's unclear, what the tolerance here is, or limitation of tolerance, so that virus loads eventually go down. Okay. So this is where Katrin and Peter from the lab started their journey, many years long journey, trying to figure out in the LCMB model whether there are limitations to this neonatal tolerance that all the textbooks are writing about. So they did initially a very simple experiment. They inoculated newborn mice that were either wild type, or they had, what I introduced to you before, a restriction in their B cell repertoire. So they had a monoclonal receptor for an unrelated antigen. And then they followed these animals into adult life. And what they saw is that the wild type animals mounted an antibody response to the viral envelope. So including determinants which can be neutralizing. They didn't have neutralizing responses, but they bound, and the antibodies bound to the outside of the virus. Something that was absent, obviously, in the monoclonal antibody mice, which only can make irrelevant specificity antibodies. So far, no surprises. What was really intriguing was that there was a viral load difference. So in liver, but as well in spleen, there was a half to 1 log difference in viral loads, depending on whether the animal was able to mount a specific antibody response to the virus that was persisting lifelong. So this is by infectious titer. Here you see it by TACMAN RT-PCR. You can even appreciate it in histological slides. I mean, here the wild type, you can see the infected cells in brown. The infection density is lower, I would say, than in the antibody incapacitated mice. This was clear, but in order to study this phenomenon even better, we turned to a attenuated strain of LCMV, a variant that we called ART-LCMV, and did the same experiment again. We again got these binding antibodies to even higher extent. Again, the B-cell receptor restricted mice couldn't do this. Some of these animals even have neutralizing antibodies, the wild types, and the differences in viral loads in the organs were absolutely astonishing, particularly in the liver. And you can appreciate that in a section. You've got these huge patches of infected cells in the B-cell receptor repertoire restricted animals, and hardly, maybe a few individual infected cells left in the wild type animals. So this virus, although given in very young, meaning first 24 hours, very young age, is eventually brought under control. So similar to what the hepatitis B carrier would do over 50 or 60 years of life. That, of course, was interesting. It allowed us to potentially study what the correlates of this immunoactive phase and viral load suppression in congenital infection are. All right, so I showed you these antibody responses, even here some neutralizing titers. I've been investigating B-cell responses to LCMV for a long time, and I can tell you they are strictly CD4 T-cell help dependent. Very much so. They often need to mature a lot, affinity, mature. So I couldn't believe that these responses would occur without cognate help. So to try and gauge if there was some cognate help around, we used such animals that had been infected at birth or controls been infected in adult life. We tested them by giving them specific B cells. We transfused them with an indicated population of congenitally marked antiviral B cells, and we checked what the B cells would do. 3 weeks after infusion, we took their spleens and stained, and we saw that they formed beautiful germinal center reactions. So you can see here the PNA in green, the transferred red cells, very densely populating these germinal centers. So I must say, that smells very much like specific help, right? How could the cells get there if nobody helps them? Can we find the cells? So, okay, we got our MHC class 2 tetramers from the NIH Tetramer Core, to acknowledge here, and we tried to stain them. And so our positive control, so there's a CD44 versus tetramer, and positive control has a nice population of specific CD4 cells, then background levels in the uninfected animal. Yeah, I think this is a hard sale, right? You're not going to buy this, that you've got some cells here in the wild type infected animal. Maybe this animal with an ART-LCMV infection, you'd say this is a little lower background, but nah, not convincing. So what do? You do magnetic bead enrichment. I hope that this is more convincing. So, in both types of carriers, we can enrich a population that we cannot enrich in the uninfected animals, meaning that there is a population, tiny, but it's there, of virus epitope specific CD4 T cells that, despite supposed tolerance, high viral loads, infection in the first 24 hours of life, these cells are present. What is their phenotype? Well, by flow, we can tell that about a quarter to a third of them has a T follicular helper phenotype, PD1, 6, 5. So yeah, that fits with the general centers we saw. We then went on and did some more deep characterization. more modern day type of assessment by single cell RNA-seq. We identified many subsets, GC, non-GC TFAges, cytotoxic, EMS high, and so forth. And this is, of course, the combined analysis, but we found for both conditions, adult infected, neonatal infected, we found the cells in every cluster. So there was no cluster that was only present in one or the other. We then also looked at specifically the GCTFages and non-GCTFages from the neonatal infected, the adult infected. And sorry, I don't think there's a real difference here. I mean, GCTFH, non-GC, yes. But the two groups, to me, they're very, very similar. So you get cells of the very same phenotype transcriptome when you have neonatal infection, just numerically, much reduced. Repetition in these subsets is a little different. I agree. You got significantly more non-GCTFH in the adult infected. You get more cytotoxic. Yeah, there is more inflammation. But by and large, these responses look pretty much similar to me. What was different, though, and now becomes more exciting, is when we did TCR sequencing from these cells and then did clonotype assessments. Very consistently, adult animals had extremely diverse repertoires of clones. So you see here the very low frequency clones, below 1 in 100 cells, they made up for the majority. So every bar is one animal, the majority of the repertoire of the adult infected animals. So many, many different clones. In the neonatal infected animals, you've got some with, having a large proportion made up of cells at very high abundance. So, few clones make up for the majority of the response. Now you can say, well, it looks alike, but can you put a number on this? Well, yeah, you can do such plots, and you can calculate Gini-Simpson index. So here what you do is you plot by ascending frequency the proportion of cells as a function of number of clonotypes. And in a perfectly homogeneously distributed population, your line should go like this, because every clone would have the same proportional representation. So it would be a straight line. But the more it deviates to the bottom right, the more sort of you have a biased repertoire. And you can see that the neonatal infected, they have much more bias than the adult infected, and you can put a p-value on this for an index. So what we see is we see far fewer cells, and these few cells are proportionally much more oligoclonal in the neonatal infected mice than the adult infected mice. Okay, so we get this B-cell mediated suppression of viral loads, or even almost clearance. So this is a viremia curve, just to show with RT-LCMV, we have suppression of viremia in a wild-type animal that goes close to detection limits of our assays. What is required for this? So, for example, we used AID, activation-induced cytokine deaminase knockouts. These animals fail to suppress viremia long term. They also fail to make good antibody titers. When we have no MHC class 2 or no CD40 ligands, so classical TB interaction partner molecules, again, viral load control fails. The wild types suppress viral loads. When you have no IL-21 receptor, or if you lack BCL6 in T cells, so a widely used model for T follicular helper cell deficiency, then also viral loads stay high long term. And you can also see this by histology. Now, the absolute density varies, but you've always the wild types almost devoid of infected cells, and then knockouts that have, some density of infected cell clusters in their livers. So this is pretty much a canonical B-cell response, it seems. But you can say, fine, so the bottle is half full in these carriers, but isn't it perhaps also half empty? And we try to test this, saying, is maybe CD4 help rate limiting for B-cell response in congenital infection? And well, to tell you it is, but how do we test this? So we infected animals at birth, and then, 8 weeks later, when they were adults, carriers, so again, these animals are full of virus from head to toe, we transferred, again, the anti-LCMV B cells, monoclonal indicator cells, the ones that I've shown you in the Germinal Center before, or we transferred them together with specific CD4 T cells, smarter cells, some of you may know them. And we did the same game in adult infection as well. So B cells alone or a combo of specific B cells and CD4s. And then we checked how many germinal center cells we got out. And it was clear. The neonatally infected mice, the specific B cells expanded, but they expanded much more, 6-fold more, when there were additional exogenously supplied CD4 cells. In the adult infected animals, the B cells alone or the B cells plus CD4s didn't make really a difference. So because there, the added CD4s are a drop into the ocean. They have so much CD4 endogenously produced, they don't need transferred smarter cells. The neonatally infected mice, they benefit very much. You see the increase. Does that translate into antibody quality? Yes, it does. So we have means to assess affinity matured antibody produced specifically by the transferred B cells. And you see that these antibodies also are about 6-fold increased. Again, adult infected animals getting the B cells or B cells with the CD4 cells, there's no difference. So the adults, they have enough help, numerical and functional. They don't need help, but the neonatally infected, they benefit from being given help. So that brings me to my final conclusions. There is a key role for the antibody response to congenital infection, and I'm referring to specifically to perinatal infection here, for viral load control. So we do think that the antibodies observed in the immunoactive phase of hepatitis B are not just biomarkers, but they are probably causally linked to viral load suppression. Not to say they were the only mediators, but based on the LCV model, we think they're really important. These antibodies, at least in our model, are strictly CD4 T cell dependent, and these CD4 cells are around. They're not completely tolerized, but they're partially, meaning there is a functional TFH response mounted, but giving exogenous more CD4 helps. And we know this response is not only numerically restricted, but also clonally restricted. So we think that there is some pruning in the thymus of specific clones because they encounter the virus and therefore are eliminated. But some cells come through, and these cells then nurture the B cell response for C. So taking this together, we think that the antiviral TFH response in prenatally infected animals, or presumably humans, is an attractive target for a functional cure. Because if, as we and others have seen, there is hardly any CD8 response, virtually none, it's very hard to blow that up with immunotherapy. But having a functional CD4 response maybe is a starting point to improve things. And particularly if these responses are limited, then we might have something to work on. Then the most important slide for last. I'd like to acknowledge all the contributors. I mentioned the protagonists, Lena Kastner, Katrin Martin, Peter Reuter. Of course, all the present members of my lab, many former members, colleagues, bioinformaticians, mouse generators in Basel University, but also from many other places. All the histology you've seen was done by Dora Merkel's team in Geneva. We work with Max Learning's team very intensely, with Sanjeev Luther, with many others in many institutions, and of course, the funding bodies for their help and support, essential help, particularly appreciated these days when it's becoming rate limiting. And thank you all for your attention and for listening in.